All right. So it's a great pleasure for me to introduce Dr. Miles Xi and Angelos Vasilopoulos. Dr. Miles Xi is an assistant professor in the Department of Mathematics and Statistics at Loyola University Chicago. He received a PhD in statistics from UCLA 2 years ago. His research focuses on developing statistical and machine learning methods for large-scale genomics data. Dr. Xi's work has practical application in biopharmaceutical statistics and electronic health records. He's also interested in applying artificial intelligence to social science. So Angelos Vasilopoulos is a senior student in statistics at Loyola University of Chicago. His research interests include model optimization, clinical outcomes modeling. He has published 2 peer-reviewed papers under the supervision of Dr. Miles. So they also participate at the most recent Data Science Week, and it's a great pleasure to have them presenting as seminar seniors. And thanks a lot also for your flexibility given the inclement weather. So thanks a lot for your presence here.
Right. So thank you, Alessandro, for your introduction. So today, me and Angelos will introduce some of our work recently about using different tools for analyzing large-scale social media and biomedical data. So first, I want to give some overview about this presentation. So basically, we will divide the whole content into 3 parts. The first one is to introduce some 2 applications using predictive models to understand political ideology from social media data. Angelos will introduce predicting the survival of tongue cancer patients. The second part is to introduce some benchmark study in large-scale single-cell RNA sequencing data, and specifically one application in data quality control, which is doublet detection. And Angelos will introduce the hyperparameter tuning of one method we find better perform in our benchmark study. And lastly, I will introduce my one methodology development, which is diversity preserving subsampling for big data analysis. So first, let's introduce our first topic, which is using predict model to understand political ideology from social media images. So the whole motivation of this study can be demonstrated by these 2 images. So as you can see here, we have 2 images from 2 very popular figures in both sides in political spectrum. So the left one is AOC, and the right one is Marjorie Taylor Greene, right? So as you can see here, if you compare these 2 pictures actually posed by themselves, you can see there's a lot of difference in terms of the political communication they want to show and the idea they want to express. So for example, in the left, you have a minority and an urban area. on the right, you have some veteran and maybe suburb area, and a lot of other things. So from those examples, we want to ask several questions. The first one is, what are the difference between liberal and conservative in terms of images they post on social media? And the second is, what do these differences mean? And the third, technically, how can we decode those information from images? Now, to answer those questions, we collect data from social media, specifically Facebook. So we collect about 20,000 images from US federal legislators, from 1/14th US Congress. So we divide them into 2 groups. There are about 68 Democrat legislators, so and 150 images per person. And also we have 68 Republican legislators and also 150 images per person. So all those images are actually downloaded from their own website, their own account on Facebook. So then we use convolutional neural network and some computer vision technique to analyze those images. So basically, the convolutional neural network is a specifically supervised learning tool to utilize spatial patterns in images. And at the end of the day, it will create embeddings with rich visual content. And we implement this CNN by PyTorch deep learning framework. So the general technical idea is that first, for the image input, you use this so-called kernel operation to catch all those spatial features and then project them into the second layer. And then you use this so-called pooling operation to catch the local maximum of those features to generate the input for the next layer. And then you repeat this kernel plus pooling by many, many times. And then at the last layer, you get one-dimensional vector with rich visual content. Now, using this tool, we did classification about party affiliation on those images. So first, we input all our images into our model and train the model using backpropagation. And we use one variant of CNN, which is ResNet34, to predict or classify the binary party affiliation. Then we use a 10-fold cross-validation to show the model classification accuracy. We find that this is about 60% classification accuracy if we check the image prediction. Then if we do a majority vote for the image from one politician, and we will find 83% classification accuracy at politician level. So we actually upload those images to Amazon Mechanical Turk to let some human testers to classify those images. And we got similar performance compared with human performance with comparing human performance with our model classification. So now here is some top model predicted liberal images. So here, all those images are classified as Democrats, and all those bounding boxes are the true party affiliation. So you see, this one is a wrong prediction, and this one also a wrong prediction. So the model thinks this image is from Democrats, but this one is actually from Republican. The possible reason is maybe there's some NATO stuff in this images, so the model thinks this one should be Democrat. And also this one is Republican, but model falsely believes this is a Democrat. Maybe it's because there's some minority children in this figure. So this is the top model predicted conservative images. So again, the bounding box are the true party affiliation. So there are some wrong predictions. For example, here, these images, they are actually liberal Democrats, but the model thinks they are Republicans. Also, the reason possibility is from this national flag. And also this one, the model thinks this is Republican, but actually this is liberal, possibly because this is a white man with a national flag. Okay. Now we also compare our predicted party probability with the ground truth ideology index. So we pick up this nominate score that's well established in political science literature and compare this score versus the model output probability of party affiliation. And we actually find there's a strong correlation between the model prediction probability and the ground truth nominate score. So as you can see here, the correlation is about 0.7. Now we also did some feature analysis, try to see is there any visual feature that models specifically pay attention to. So the first three show three liberal images, and all those colorful parts are where the model focus on to make decisions. So those features are captured by this gradient-based localization. So basically, you take gradient of your model output with respect to the original pixel, and which gradient is higher will be assigned at all those colorful areas. So as you can see here, for those liberal images, the model focus on more on those like forest and some gender minorities and also some racial minorities. And for those Republican images, you can see the model is more focused on this like military uniform, national flag, and also this uniform, this suit. Maybe they represent some businessman. So basically here, the model is making decisions similar to human in terms of the political ideology about the party affiliation. Now, next, we did some facial expression analysis. We basically use face detection technique to catch all the faces from the politician images, and then we use a public available facial data set to train a classification model with those 7 different type of facial expression. And we apply the model to classify the facial expression on our politician images. And here is the model prediction result. So basically, we have angry, disgust, fear, happy, sad, surprise, and neutral. So this is one prediction example. So here we summarize those facial expression distribution and text to compare the difference. So this figure, left one, shows the facial expression of main legislators, the politicians themselves. And the right one shows the facial expression of the associates in their images. So people know themselves. So we find that basically both parties will have the happiness as the dominant facial expression in all their images and followed by neutral facial expression. However, there are significant difference between Republican and the Democrats in terms of expression. So specifically, Republican actually express more happiness than their Democrat counterpart, right? So as you can see here, the Republican has higher ratio of happiness for themselves and also for the associates in their images. We also did a racial and a gender distribution analysis. So similarly, we train a model on public data set with race and gender labels, and we apply the model on our facial images to see what's the distribution and the difference in terms of gender and race in these two party affiliation images. So we find that the liberal images actually contains more female and more minority groups. So for example, here you can see the Democrats has less white, but more Black, Asian, Indian, and others. And also the Republican has more white and also more male in their images. So lastly, we also did object analysis. So here we use objective object detection tool from Google Vision API to catch all those objects from the images. And then here we show the top 10 Democrat associated objects and the top 10 Republican associated objects. As you can see here, in Democrat objects, we have more like natural things like sea, shore, coast, and like so to show that maybe there are more environmental friendly ideology here. And for Republican images, we find more like military and uniform troop and senior called something. So that probably reflects their ideology in terms of those objects. Okay, so basically to summarize this study, So we actually find that the social media images posted by these two party politicians actually is a very strong indicator for their political ideology. So basically, for conservatives, they have more happiness face in their images and have more white, male, and the military, law, or senior related content. For liberal, on the other hand, they have more unhappiness, minority, female, and natural environment related images. So those conclusions actually, those observations actually, they are consistent with the classical like expression for those two parties. And so also this type of framework is is actually an emerging field which is called computational social science. So different from the traditional table data, we use unstructured data, including images and text, plus web and social media to analyze some traditional questions in political science and social science. So this is the first study we want to introduce using predictive model. And the second one is to use a predictive model to predict the survival of tongue cancer patients. So I will hand over to Angelus for this part. So I think I need to stop sharing, okay?
As you change to the other one, I just wanted to say it's interesting to wonder if you could create a model where you plug in a picture of a person, and then the model tells you, and then the model tells you that person's political affiliation just based on like their facial expression, for example, or you know,
based on facial expression. So
I mean, maybe one picture wouldn't be enough, but it would be interesting to put in a person's entire social media profile and then see what their political affiliation is.
Yeah, I think that would be a stronger model because you include both image and text and plus some structural data, right? I think it's called a multi-module model. So here, but here all we focus on the only the image they post by themselves, and also those images is not only about the face. They are also about some other things, not only about the human, right? So like, so if you see here, you have human, but also you have some other stuff, right? So this is no human here. This is a building, right? So
like a flag,
right?
Yeah.
Yep.
All right. Should I share my screen?
Yes, please.
Yeah, feel free to share your screen. Yeah.
Can you see my screen?
Yeah.
Yes, but just in part. Just in part. It's yeah. Try to exactly. So let's see what happens when you click presenter view.
All right.
Yeah.
Oh, I'm on the wrong slide. Just a second.
This is the other part.
Right. Okay, here we are. So the following presentation, as Dr. Xi mentioned, is about predicting the survival of tongue cancer surgery recipients by machine learning models. As you may already know, tongue cancer is a frequently diagnosed head and neck malignancy, and the ability to predict survival of this malignancy touches on is very important for making treatment decisions. So this is where machine learning comes in. Machine learning could be effective for predicting survival from patient data. Currently, there aren't many machine learning models of tongue cancer survival in the literature. Some risk factors have been determined in some studies, but there aren't many models. There aren't many models of the nonlinear relationships of survival to these risk factors. Our objective in this analysis is to predict tongue cancer surgery recipient survival from a clinical data set of patients of that interest group. So this data set that we're working with was collected at Changgung Memorial Hospital in Taiwan. by Tsai et al. They've also written a survival analysis paper using this data set, which is also interesting to read. But again, our analysis is more machine learning, explores some different methods. And the data set was collected from 2004 to 2013. It includes 1712 patients total, 1,712 patients total. 1,280 of them survive, and 432 of them do not survive. So as you will see, and this is something important, this data set is not balanced, and there are a few things that we do about that in our analysis. So the variables included in this analysis are patient age, gender, AJCC stage, T stage, N stage, and grade, and whether radiation therapy and chemotherapy were received by a patient. There's also the survival outcome variable, and then there were several other variables as well, but these were removed for various reasons. For example, site of operation was a non-variable. So in other words, all operations were on the tongue for every observation, for every patient. And so that variable was removed because it didn't offer any explanatory. It didn't have any explanatory power. Operation occurrence was also a non-variable since all patients in this data set were operated on. And then follow-up time was also removed from our analysis. And the reason for this was that follow-up time isn't known until long after the time of an operation. So it's not usable in practice for prediction. One of the first predictive models that we constructed was a k-nearest neighbors model. A k-nearest neighbors model essentially works by labeling observations according to Euclidean distance from other observations. We also trained and tested a random forest model. A random forest model makes predictions according to a majority vote of a randomly generated set of decision trees. So a specified number of decision trees are generated, and then those decision trees each decision tree produces a set of outcomes, and then whatever most decision trees vote on, so to speak, is the decision of the entire forest, is the decision set of the forest. Extreme gradient boosting, or XGBoost, works in a similar way, but decision trees are generated sequentially, and each new tree learns from the errors or residuals of the previous trees. We also attempted prediction using logistic lasso regression, which is a kind of regularized regression. And this kind of regularized regression introduces a regularization parameter that effectively eliminates some model predictors and gives some interesting results. Finally, we use these four models: k-nearest neighbors, random forest, extreme gradient boosting, and logistic lasso regression to create a fifth ensemble model. An ensemble model in general is constructed in different ways, but in our case, we used a fairly simple scheme of averaging outcome probabilities across models. A large part of this project, and well, you'll see the outcomes of these models in the results of these models in just a second, but a large part of this project was model optimization. So according to our analysis, these were the optimal hyperparameters according to the area under the precision recall curve. It's important to note that the cross-validation scheme that we used to find these values wasn't nested, so these values are likely biased to our data set. However, the purpose of our analysis isn't to make a recommendation for a specific recommendation for the best hyperparameters, but to suggest an analytical framework for future attempts to predict tongue cancer surgery recipient survival. So after optimization of our models, we measured their performance as accuracy, area under the precision recall curve, precision recall, TNR, and balanced accuracy. The extreme gradient boosting model achieved the highest accuracy, precision, TNR, and balanced accuracy. So we might be tempted to say that it was overall the best model, while random forest achieved the highest recall and the ensemble model achieved the highest AUPRC. So again, AUPRC was our optimization criteria, but model hyperparameters can be determined with different criteria depending on the optimization objective. It's also interesting to note that the low true negative rates achieved by all models. Interestingly, TNR increases when changing the cutoff probability value from the default 0.5. And while this is generally at a cost to other metrics, it does speak to the flexibility of these 5 models in predicting positives and negatives, so predicting survival and non-survival. We measured the uncertainty of these performance measurements by bootstrapping. So these are basically our prediction performance measurements. and this is their uncertainty. So it's always good to, to measure the uncertainty, to estimate the uncertainty of a measurement. In our case, we did this by bootstrapping, and you can see the results in these figures. You can look at these more closely in our paper, which I will touch on in just a second, which I will reference in just a second. Another important part of our analysis was feature selection, which was done according to permutation feature importance. In our study, permutation feature importance was the decrease in AUPRC that resulted from permuting or scrambling the values of a variable so that the relationship between the variable and survival was broken. So to understand permutation feature importance, a good way to think about it is that it's iterated as many times, the same number of times as you have variables. So in each iteration in each permutation, one variable in the data set is permuted or scrambled so that it basically becomes just a bunch of random numbers. And then this permuted variable is used in building a model. And if we see that the performance of the model doesn't change, even though we've scrambled the variable, it means that the variable doesn't actually contribute anything important to the model. And then the model has low permutation feature importance. And then that's justification to remove it from the model building. So these are the results. So again, these are the results of the permutation feature importance calculation. So dividing feature importance by the highest variable importance gives normalized importance, which you can see in this figure. And as you can see in the figure, tumor grade contributes the most to prediction of patient survival, followed by end-stage, T-stage, chemotherapy, and radiation therapy. And these variables are all consistent with previous findings in clinical and modeling studies. For example, histological grading and the TNM staging system of the AJCC, which includes tumor grade, end stage, and T stage, are pretty well established prognostic factors in oral cancer diagnosis and treatment. And numerous studies also suggest the importance of adjuvant therapy like chemotherapy and radiation therapy, especially for patients in advanced stages. On the other hand, age and gender are among the least important variables in the model prediction, making less than 10% of the contribution of the top variable, tumor grade. These two variables are also found to have less impact on patient survival in clinical studies, although it's important to wonder how this permutation feature importance order would change if we were focusing solely on a pediatric population, as the population in this study was mostly older. In the final stage of our analysis, we wanted to assess the calibration of our models. In other words, their tendency to produce false positives and false negatives. We visualize this with a series of calibration curves, which you can see on this slide. And so just to understand these curves, points above the diagonal dotted line indicate false negatives. So random forest, for example, had many false negatives, as you can see in the second graph on the top row, while XGBoost was the best calibrated model. Finally, since we were dealing with an imbalanced data set, as I mentioned at the beginning, we wanted to explore how balancing the data set could affect our results. As you can see in this figure, while the raw data set resulted in the highest accuracy and recall, undersampling positive observations resulted in undersampling meaning multiplying a certain subset of observations so that they have a higher weight in the data set. So this kind of undersampling of positive observations resulted in the highest precision in TNR, while oversampling negative observations resulted in the highest balanced accuracy, and oversampling negatives and undersampling positives, which is a hybrid sampling scheme, resulted in the highest AUPRC, which again was our original optimization criteria. Clearly, there is some benefit to changing sampling schemes, which may be a very interesting direction, which may be very interesting as a future direction. Thank you so much for listening to our presentation, and you can learn more about this analysis by reading our paper recently published in Advances in Artificial Intelligence and Machine Learning. And now I will hand it off to Dr. Xi for the next part of the presentation. Thank you again.
Okay, thank you, Angelos. I will share my screen again to introduce our second part, which is some benchmark studying on single-cell RNA sequencing data. So yes, as you can see here, So here I want to start a new topic about single-cell RNA sequencing data and the doublet detection issue in this specific data format. So now, before that, I want to introduce some general background, which is central dogma of molecular biology. So as we all know, in human and animal bodies, actually our genomic information is stored in this DNA strands. So DNA is a double-stranded nucleotide sequence, all those ACGT sequences. And at the same time, the genomic information can be also found in RNA sequence, which is a single-stranded nucleotide sequence. Now, the central dogma of molecular biology told us that the genomic information is coming from DNA and transcribed into RNA and finally implemented in protein. So the information flow is from DNA to RNA and protein. So there are many different techniques to quantify different genomic information at different levels. So in our study, we focus on this RNA level because the RNA transcribed is a direct predecessor of protein. You may have same DNA in different bodies, but the transcription in RNA will be very different. So then you will have very different proteins, which is basically shown in reality. So now the single-cell RNA sequencing technique is try to quantify the gene expression in RNA. It is emerging sequencing technique that review genome-wide gene expression at single-cell levels. It can measure the expression of tens of thousands genes in millions of cells, and it has a wide applications in cancer therapy, drug discovery, vaccine development, and precision medicine. So here is the general process of doing experiment and generate single-cell RNA sequencing data. So the most important part is to isolate all single cells and label each cell. And then you can extract those labeled RNA and amplify them into amplified RNA, and then do sequencing, map them back to your gene library to know how many genes in each cell is expressed. So the data generated from this process has several challenges for data analysis. For example, it is very high dimensional, which means you may have more than 20,000 dimensions and up to more than millions of sample size. And also, the data matrix generated is very sparse and noisy. So you may have more than 90% zeros in this final data matrix. So here is one example of applying RNA-seq to medicine. So the first figure shows that you can do single-cell RNA sequencing on cancer cells and healthy cells. And then you generate these two data matrix, and you can compare the genes in these two matrices. For example, you can do differential expression gene analysis. If you find gene 1 is highly expressed in cancer cells, then you may identify some biomarkers that contribute more to this type of cancer. And the second example shows how you can design and test a drug to solve this problem, which is you design a drug to depress the expression of gene 1, try to solve this cancer problem. And to test the efficacy of your drug, you can do RNA-seq on both cell after applying drug and cell after applying placebo. And if you can test the gene 1, try to see if they are really depressed by your drug. And by doing that, you can know the efficacy of your drug. So there's many issues and the challenges in single-cell RNA sequencing data analysis. So we want to focus on one specific problem, which is called doublet. So in single-cell RNA sequencing, analysis. So each cell is captured in one reaction volume to give you a singlet. So for example, here you have this vertical bead go through these pipes, and then you have this horizontal bead go through this pipe, and the vertical cell flows go through this way. So then each bead will catch single cell and get some labels. You will have singlet. However, you may have some so-called doublets formed when two or more cells are encapsulated in one reaction volume. So, and the potential doublet rate can be up to more than 40% in some data sets. So the formation of doublets are a major confounder in downstream data analysis because they are not real cells, but they are treated as real cells. So, and there are three examples of the bad effect of doublet formation. For example, if you want to cluster your cells into different clusters, you may have spatial cell clusters. So the doublets themselves will give you a cluster, which shouldn't happen because the doublets do not exist in your data set. Or in your trajectory analysis, this is the true trajectory. However, the doublets may connect these two branches to give you a bridge. And also, it can bias the identification of differentially expression genes. So to solve this problem, actually, so far, there are about 10 computational doublet detection methods have been developed to detect doublets from data. However, there's no guidance to choose among all those methods. And to address this issue, we conduct the first ever comprehensive benchmark study in this field. So basically, we collect a large-scale real and simulated data and apply all those methods on or design the framework. So basically, we use overall detection accuracy, three major downstream analyses, and also computational efficiency and the software quality to evaluate all those 9 major methods and try to provide a guidance to improve the existing methods and also for biologists to choose their best method for their specific task. So, so I will skip some technical detail, but at the end of the day, we find this ICDBL finder method actually shows the best detection accuracy and high computational efficiency. And we developed our package doublet collection actually to integrate all those different methods for users to more conveniently use different methods. And also we post our real and simulated data sets for further method evaluation and development. So next, Angelus will introduce our hyperparameter tuning strategy for this best method ICDBL finder to further improve the performance of this doublet detection. So I will hand over back to Angelus to let him to introduce this topic.
Okay, can you see my screen?
Yes.
Yes, very well.
All right. So the method that we focused on in our study was SCDBL finder, which, as Dr. Xi mentioned, is one of the doublet detection algorithms. So SCDBL finder, like most functions, or like the functions of SCDBL finder, like most of the functions in R and in many other programming languages, comes with a set of user-specified hyperparameters. These hyperparameters determine the specifications of the data processing and scoring procedures. So in our analysis, we decided to focus on the hyperparameters NF, which is the number of genes considered in the analysis, PC, the number of principal components used for classification after dimension reduction, and depth, which is the depth of the trees used in the gradient boosting labeling procedure. So in the last column of the table are listed the hyperparameter values that we were interested in testing. These values were selected based on best practices as reported in the literature, and you can read more about these best practices in our paper, Tuning Hyperparameters of Doublet Detection Methods for Single-Cell RNA Sequencing Data, which is also referenced at the end of this presentation. In total, we tested 125 hyperparameter combinations as an initial grid search. As expected, each combination resulted in a different doublet detection accuracy, which we measured as area under the precision recall curve. Results per data set are summarized in this table. We also measured true positive rate and true negative rate per data set, which you can see in the following two slides. So again, you can look at these more closely in our paper, which I will reference again at the end. And we modeled the relationship after determining AUPRC. We modeled the relationship between AUPRC and different hyperparameter values by second-degree polynomial regression, including in regression NF, PC, and depth as the first and second at the first and second orders and their two-way interactions. So we were basically building a simple linear regression model between to model the relationship and find the pattern between AUPRC and the different hyperparameter values. So a t-test or a series of t-tests suggested that NF and PC are significantly associated with area under the precision recall curve, yielding the following final model of AUPRC, which you can see on this slide. Some calculus brings us to the conclusion that the optimal hyperparameters rounded up to the nearest integers are NF of 1352 and PC of 19. So in other words, the overall most accurate doublet detection results from using 1,352 genes or variables and the first 19 principal components. However, it is important to understand that these values are optimal over the 16 data sets included in this analysis, and we do have to consider whether they are optimal for individual data sets. So as it turns out, to some extent they are. As you can see overall in this figure, overall optimal hyperparameters resulted in more accurate doublet detection in all but 4 data sets, with AUPRCs of 10 data sets ranking at the 20th percentile or higher, and all data sets achieving the 50th percentile or higher. Now, to explore the generalizability of our optimization results, we performed optimization again but this time within cell type groups. So in other words, we determined optimal hyperparameters for data sets in the same cell types, namely peripheral blood mononuclear cells and in human embryonic kidney cells. As expected, hyperparameters were different per cell type group and overall, speaking to the importance of considering biological factors when implementing our hyperparameter optimization procedure. The key takeaways of this analysis are the following. First, there are no universally optimal hyperparameters, and this is something that's true almost generally. Yet, hyperparameters can be optimized per specific analytical objectives and per biological conditions. Thank you so much for your attention and for listening to this presentation. I'll turn it back to Dr. Xi and Dr. Selviteja.
Yes, thank you, Angela. So we have one topic left. I will quickly go through that. Also, I want to mention that this optimization.
Yes, you have time. You have time.
Yeah,
so
yeah. Yeah, so yeah, I want to mention that this optimization work has already been accepted by quantitative biology, so we'll be forthcoming shortly. So last topic we want to introduce is diversity preserving subsampling for big data. So this part is basically this work has partially been published. The first part has already been published. And Andres and I, we are working on the improvement of this part and try to get published maybe in a couple of months. So now, the motivation of this study is a big data challenge. So a remarkable achievement of modern data science is the large-scale data set is generated. For example, you may have up to 1 million data points and thousands of features. So in our previous analysis, we actually have this Cortex single-cell RNA sequencing data contains 1.1 million cells and more than 2,000 genes. And also in our social media analysis, some public data sets actually contain more than 100 million of high-resolution images. So to analyze this data set, usually you have to do some subsampling to collect a small set from the big data. The reason is that you may need to do a quick exploratory data analysis, for example, visualization, or you may need to construct some data set for specific purpose. For example, if we want to classify different human facial expression, then we need to pick up a small subset from this huge data set with human images to train our model. So a straightforward solution to collect a good subset from big data is to randomly select a small subsample from the full data. So however, random subsampling usually we will miss some small subtests and also fail to preserve the diversity of your original data. So here are two examples. For example, in this single-cell RNA sequencing data, the original full data actually gives you 3 patterns. You have a trajectory here and a bigger cluster and a small cluster, which is a rare cell type. Now, by randomly subsampling, you may miss this trajectory pattern, or you may have very small number of rare cell types. Then the random subsample may not give you the original rich information from your data. Now, the second example is basically shows several distributions from different data sets in terms of their racial distribution. So those data sets are all from public database with some human faces. As you can see here, because the original source of this data set is biased toward, say, the white race group, and you have very small number of minority group in all those sources. Therefore, by using randomly subsampling, the data set they created actually will bias towards the majority racial group, and then the model trained on this data will have very light accuracy to classify the minority group. So to solve those problems by randomly subsampling, we developed a IC sampler for faster diversity preserving subsampling from big data. So the idea is that we want to make the data points in our subsample as separative as possible to represent the diversity in the full data. So here is one example. Originally, you have a full data with 3 structures, then the random sampling will miss many information. But the diversity or IC sampler can achieve this diversity preserving by put more emphasis on these 2 special structures, such that you can also detect this pattern from a small subsample. So the essential idea of IC sampler is using a sequential selection algorithm. So the idea is that the first step is to randomly choose a data point x1 from the full data, and then choose the second x2 up to the xns. So ns is the size of your subsample. Then in every step, you will do this. So for suppose you already have m data points in the current subsample, then the next m+1 data point is selected by this criteria. So in this formula, you want to choose the new data point, minimizing this ratio. So here in this formula, the d is the Euclidean distance between the current data point and your new data point. And the f function is the kernel density of each data point. and alpha is a constant. So by using this criteria, you choose the other one, the next data point, until you finish every selection. So here I want to give more detail about why we want to use this minimization to choose our data point. As you can see here, this minimization is a summation across these ratios. And in the numerator, you have the product of kernel density between your new data point and all the current data points. And in denominator, you have a Euclidean distance between your new data point and the current data point. So these criteria can achieve three objectives. So minimizing this means that you should maximize this Euclidean distance, which means the data points in the subsample are as separative as possible. And also the numerator shows that minimize the whole term will minimize the density function of your data point, which means that you want to choose the data point with very small density. So you give more priority to small subgroups in the full data. Using these two, according to these two achievements, finally, after you finish the algorithm, we will obtain a diverse subsample. So here is one numerical result to show how the method performs. So the left figure shows the random subsample from a big data. And this one is our method. So all those gray areas are the two-dimensional plot of your original big data. And the red one is the data point chosen by two sampling method, random and subsample. As you can see here, the IC sampler or method actually choose more data points and cells from those relative small group and put less emphasis on this larger group. However, for the random pickup, you will pick like regarding this of all the original distribution of your data. So you will miss all those small subsets. But in our selection, we emphasize collect more those small subgroups. So here, why this selection works. So the previous work actually shows that our sequential algorithm is essentially minimizing the hot-off distance between subsample and full data. So what is hot-off distance? So hot-off distance measures the similarity between two sets. It is defined as the largest of all distances from a point in one set to its closest point in the other set. So in other words, for every, so there's two sets, X and Y. For every point in Y, you first find the smallest distance from this dxy and one data point here. And for other data points, you also find the smallest distance. And among all those smallest distances, you find the largest one, the smallest distance as your hot-off distance, which is this max-min distance. Okay, so the closest is internal, and maximum is applying on all those distances. So actually, we show that our algorithm can achieve minimization of hot-off distance between subsample and full data. However, if you want to achieve this minimization initially, you have to do a very huge computation, which is actually infeasible in large data sets. For example, the time complexity of a brute force search to get a minimization of hot-off distance is this large. So you have to choose you have a combination here and times the sample size and the force size, which is not doable. So our method, because it is a sequential selection, so it is a greedy algorithm and it reduces the computational complexity to only this NS times N. So we can drop this combination because we don't have to enumerate all the combinations, NS data points from your large data. So we drop this one. We only do a sequential selection. So the only computation burden is to calculate the pairwise distance. And to further accelerate this algorithm, we split the data into different B subsets. So, and we do sequential selection separately in each subset. So this trick can improve the computational efficiency because the computational time in each subset will be N times NS divided by B squared, right? Because you split by B, so every term will divide by B. So you have B squared here. Then you combine all those separate subsets and you times B. So at the end of the day, the final computation time will be N times NS divided by B. So we shrink the time by a factor of B. So here is another numerical result to show the performance. So basically, as you can see here, we have random subsampling, and two competing methods, and all methods with different number of subsets we split. So basically, the essential idea in those figures is that if we didn't split our data, then our method as a sampler will achieve the smallest cutoff distance in a decent computation time. If we increase the number of splits in our method, we dramatically improve the computational speed, like in this one. That's the fastest method. However, we still significantly outperform other methods and specifically the random subsampling. So we can give users a flexibility between the performance and the computational time. So here is some preliminary results on improvement of our subsampling on clustering. So basically, we show that the data selected by our method can achieve more balanced cell type in subsample. So for example, our Gini index in our small samples actually achieved the smallest among 90% of 8 simulated data sets with ground truth cell type. So more balanced subgroup. So here is one preliminary result about clustering method. So here we show two data sets and clustering we do k-means clustering on subsets collected by different methods. As you can see here, across different subsample size, our method actually has the highest clustering performance compared to other methods. So to summarize, our IC sampler is an unsupervised learning method that can select a representative subsampling. And essentially, it minimizes the cutoff distance between the subsample and the full data. So it can preserve the diversity from the full data, and also our sequential selection can increase the computational efficiency by a large degree. And not only our method can be applied to large-scale genomic data, but also we will show the method can also be applied to other types like image data. And also we implement the first part of our method in R and Python package. So you can check, this is our publication on this topic. Yeah, so I think this is all we want to introduce for today. And I want to thank Angelus for doing the two projects we already published. And also Angelus is also doing this clustering validation part on our method. And hopefully it will get published in a couple of months. And thank you for your attention. And I'm looking for your questions and comments. Yep.
Thanks a lot for your nice presentations to both of you. So
Yeah. Thank you.
the sound is better in person, but
Yeah. Yeah.
so yeah, I have a couple of questions. The one question is actually, let me start with the second one, which is related to the fifth project that you discussed. So so let me try to see if I understood correctly why. So the subsampling procedure. So you have a large data set that is computationally too expensive to analyze, so you do subsampling, and then to preserve diversity, the same diversity, to make the subsample representative of the original sample, you just have to you try to use a method to include the rare subsets. Is that correct?
Right. So yeah, you want to collect small subsets. However, the random sampling will not guarantee you have those small sets. And sometimes the small set is kind of important because, say, for example, for biologists, we want to collect small cell type because those rare cell type may be a new cell type. So
For example,
they want, yeah.
Yeah, so the reason for which I asked is, for example, like something like stratified sampling. Namely, you do random sampling, but for each class. Do you address it? Yeah.
Yeah, so here it's unsupervised, which means we don't know the true label of your 4 data, right? So you are blended, so there's no label here. Of course, if you have label, you can just like randomly choose across different labels. However, here we don't have labels. So for example, in biology, so biologists, they do experiments. The goal is to find cell type, right? So before you do experiment, you know nothing about, or with the data, you know nothing about the cell type.
So let me rephrase your the comment. So you base the representativeness on the fact that you have explored many regions of the sample space using that sequential algorithm that you discussed. And because you're more or less going in every point of sample space, you're satisfied with the representativeness you get, independent of the fact that you know exactly which samples you include in the subsamples, which labels you include in subsamples. Is that correct?
Yeah, that's correct.
Good.
Yeah.
Thank you.
Yeah.
So good. So the other question is related to I think the project on survival so on the tongue cancer. Yeah. So I didn't catch exactly how you do the cross validation component there.
Yeah.
It was a 5-fold cross validation, and as I mentioned, ideally we would have done a nested sampling scheme. The reason we didn't was because of the small data set on the small number. Yep, go ahead.
Maybe I'll specify a little better my question. So what do you divide in 5 in K folds, let's say? Do you divide the individuals? What do you do? How do you do that? So do you divide the group of individuals into 5 pieces, or divide something else into 5 pieces?
No, we divided all of the patients into 5 pieces.
You divide all the patients into pieces. Yes, that's it. And then what is your why there? Is it your why? Do you account for censoring? I didn't catch that. So do you account for the fact that survivals are some sort of sensor data? Do you account for that, or just predict the time?
We just predicted survival and non-survival.
Okay.
Right. The Y is binary 0-1.
Okay. Why binary 0-1? So you don't do things like account for the fact that in an experiment you might not observe let's say you want to see if even experiment for 5 years, if if one of the individuals doesn't die after the 5 years, then that observation is censored. You just count it as 0.
Yes.
Is that correct? Okay, got it. Okay, very good. All right. Yes, I guess that's the end of the seminar. Well, thanks a lot for the very interesting series of topics. It's great to have you here, even if on just online. Well, another time we're gonna meet in person, both of you. So
Yeah.
So I'm gonna stop the recording now.
Right.
Thanks a lot again.
Yeah, thank you so much. Yeah.